What is CRISPR? It's basically a way for scientists to genetically modify organisms, but how does this work? What does it stand for? And how could this technology impact our lives? First, to understand CRISPR, we need to know how information is stored within organisms. There is so much information about an organism, from hair color to bone density. In organisms, this information is stored in something called the genome. Basically, a genome is the instruction manual on how to build an organism. DNA is like the language that all the information in the genome is written in. If you compare an organism to a video game, instead of having the information stored in a memory card, it's stored in the genome. And instead of having the information being written in JavaScript, it's written in DNA. DNA looks like this, an almost ladder-shaped pattern. The long ribbons are the phosphate and sugar backbones of the DNA, where the rungs are the 4 different nitrogen bases. This is how genetic information is stored, and if these bases are placed in a different order, then the genetic information is different. Therefore, by altering specific sections of DNA, you can change the genetic information of an organism. This is where CRISPR, otherwise known as clustered regularly interspaced short palindromic repeat, comes into play. Since the beginning of time, viruses have been targeting bacteria. Usually the bacterium will die, but if it survives, it will use the CRISPR system to protect itself against future attacks. This occurs when the bacterium keeps a section of the virus's DNA inside their own genetic code, like an archive. Then, if the bacterium and the virus come into contact again, the bacterium has a way to protect itself. The bacteria copies the archived DNA of the virus into a guide RNA strand. RNA is similar to DNA, except it has a single helix and translates information into proteins. The guide RNA then arms a protein called Cas9 with the information on the virus. The protein compares the archived DNA from the bacterium to the DNA from the virus attacking the bacterium. When the Cas9 protein finds a match, the protein is then able to target specific DNA sections in the virus and cut them out. Finally, the Cas9 protein is able to repair the broken DNA by replacing the genetic information within the virus. Think of CRISPR as a pair of scissors that has the ability to cut out certain sections of the DNA and replace it with the new desired genetic information. The big scientific discovery surrounding CRISPR is that scientists can now manipulate how CRISPR works. If scientists were to give a Cas9 protein a copy of the DNA they want to manipulate, then place the CRISPR system into a living cell, the cell would then become genetically altered. As well, CRISPR can be used on all living cells, meaning all organisms can be altered, is relatively quick at altering the genomes of an organism, and is cheaper than many previous methods. So what does this mean for the future of humans? Well, the use of CRISPR to genetically modify organisms is still in its early stages, meaning I couldn't just change my eye color if I wanted to. As well, scientists aren't entirely sure what every single gene in the human genome does. However, there are experiments currently happening where CRISPR is being used to strengthen the cells within the immune system. This could mean HIV and cancer cells could no longer harm a genetically modified human. Therefore, CRISPR still has a long way to go, but it has the ability to greatly impact human life forever.